Hey guys, welcome to Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. Today, let's get introduced to one of the very common concepts that forms the major practical step in most of the life science or the biological, genetic engineering, or any cell culture laboratory. That is electrophoresis, in particularly gel electrophoresis. So this technique, it forms the basis if you want to understand and study your concept at the DNA level. It is a visualization essay which helps in understanding the nature of the DNA, identification of the certain fragments of DNA to understand whether the DNA is intact or it has been broken down. So all these things can be understood using this particular technique. So anyone who is interested in life sciences or in the research area, this technique will be very useful for you. And today I'll be discussing about some of the my own experiences that I experienced while working in the lab using gel electrophoresis technique. So let's get started with gel electrophoresis. So this particular technique, that is electrophoresis, was developed by a scientist called as Arne and he got the Nobel Prize in the year 1948 for the same technique. So he is known as the father of electrophoresis. So this particular technique is based on a very simple principle that we have been learning even from right from our lower classes. That is, opposites attract each other and the like ones, the similar ones, they will repel each other. So this forms the very basic concept of your gel electrophoresis. So this is in particularly used for studying the charged molecules. So when we know the charge on a particular molecule, whether it is negative or positive, this particular assay can be very easily developed. So suppose we have a charged molecule and if we place it in some electric field, so it is obvious that it will start moving towards the opposite side or the negative pole or the opposite pole. So this particular concept is used as the principle of gel electrophoresis or rather electrophoresis. Then proteins, they do not have any net charge, but the nucleic acids, they have negative charge, so they can be easily studied by using this particular technique. So let's see what is the definition of electrophoresis. So this technique is basically a separation technique, okay, a separation technique that is used for separation of the charged molecule in a given solution based on their tendency to move towards the electrodes. If your molecule, charged molecule is negative, definitely it will start moving towards the positive side. Whereas if your molecule, charged molecule is negative in nature, it will start moving towards the positive side. So in this way, this particular technique is called as electrophoresis. So basically the term electrophoresis, it is a combination of two terms, right? So if we have this electro and phoresis, so we have two parts of it, or we can define it by breaking it down into two words. One is electro, that is applying the electric field, and phoresis means the separation or the segregation. So from the word itself we know it is a process or it is a method of separation of charged molecules under the application or under the surveillance of the electric field. So this is the definition of electrophoresis. So biomolecules such as nucleotides, amino acids, DNA, RNA, okay, they are the charged molecules. So they can be easily separated using this particular technique. Whereas when we have molecules like sugars, which do not have charge, right, they cannot be separated using this particular technique. So let us try to understand the principle behind gel electrophoresis. Suppose now if a direct current is passed through a solution, okay, and that solution contains some charged molecule. So what will happen is the positively charged cations, they will start migrating towards the opposite, that is the cathode, and the negatively charged anions, they will start moving towards the anode. So the mobility of the molecules, okay, however, it depends on the net charge, the size and the shape of the molecule, okay, the pH and the overall composition of the medium that we have used, the buffers that we have used, okay. So these are the things which we need to keep in mind. So your DNA and RNA, they being negatively charged molecules, they will start migrating towards the anode. And small molecules are going to move faster. So for example, if there is a running race, okay, if we have a running race and if there are people standing in line, so I'm not making some generalized statement, I'm just saying, I'm just giving you one example. If we have a person, okay, they are of same, say, size and shape, everything, but let's not talk about the size of any person. So if we have 3 4 people of the same size and weight, whatever, and if we give them some loaded bags of different weights, so and if we ask them to start running towards a particular point, so definitely the person who is not having any weight or who is having some lighter weight will be able to run faster and reach the destination. And rather the person who is having or who has been loaded with lots of weight, it will be difficult for him to run and reach the final line, right? The same concept goes over here. The smaller the particle, it will have a smaller molecular size and it will be able to move faster, and hence it will reach the line or it will cover more distance as compared to the larger particle. So this basic principle is used. So it is not just the electric charge but also the size and the shape and the overall composition of the buffers and the media that we have used. All these things are going to affect the rate of electrophoresis. So this principle is used in the separation of DNA and RNA. So proteins also migrate to supporting medium based on their net electric charge at a particular pH and size. So if we have two proteins and they are having identical charge, same shape, okay, then using electrophoresis would be difficult. So for that we have some other technique, so which we will cover in some other episode. Yes. So another thing that is required here is a supporting medium or a matrix, what we call as, okay. So it is required to separate the molecules according to their size and the charge. So if an important point is it should not interact, okay, or react with that particular molecule that we are using. So that means it should be inert, right? Point number one, the supporting material that we have used, it should be inert. Second point, it should be non-toxic, because in most of the cases this particular technique, after using this technique, we should be able to remove out that particular charged molecule so that we can study it further. So the materials that we have used, it should not interfere at any given point with the nature of this charged molecule that we have used for separation, okay. Then all these things are going to affect the specific porosity, because depending on the size of the molecule we will choose the porosity, how porous or what should be the pore size of that particular material. Because now why this pore size is important? Because it should have good filtering. Okay, if your molecule is so big that it cannot even pass through that particular material you have used, right? So what is the point of doing this technique? So it will be of no use. So we should have good study about the nature of the material, the charged molecule that we want to separate, and also the nature and the chemical composition of the supporting medium. So another thing, it should not interfere with the electrical conductivity of the buffers, because we are going to use buffers, right? So we have different uh, we have different sorts of examples or options which can be used. One is we can use paper strips. Okay, so mostly it is made up of cellulose acetate. Okay, starch. Then we have another one that is called as agarose, that is the most commonly used, and it is called as gel electrophoresis. Apart from this, we have cephadox, then we have polyacrylamide. So these are some of the supporting medium which can be used for conducting electrophoresis. Alright. So next is that the pH is going to affect the movement of the molecules through the pores of that particular supporting medium. So in order to keep the medium at a constant pH, the buffers are used, because we know buffers are something that will maintain the pH, right? So mostly we use phosphate buffer, tris borate, okay, TBE what we call, that is tris borate EDTA, or TAE, that is tris acetate EDTA. These are the most commonly used. Then tris glycine is mostly used for your polyacrylamide gels. So we have variety of it which can be used for electrophoresis. Okay. So then now let us move on to the next slide with this particular background. Now, based on the type of the supporting medium or the material which is used for separating the biomolecules, we have different types of electrophoresis, and they are paper electrophoresis, agarose gel electrophoresis, which is commonly called as gel electrophoresis, then page, that is polyacrylamide gel electrophoresis. So here also we have different varieties also. Then we have sodium dodecyl sulfate polyacrylamide gel electrophoresis, which is called as SDS page. Then we have something called as native page. Okay, it is something called as native page. In native page, it is used when we want to study the intact protein molecule. And another one, that is pulse filled gel electrophoresis. So this particular method is specially used in studying certain zoonotic organisms. Okay, so it is used as a gold standard method. So I was working with one of the in one of the labs. So there they were working on a particular zoonotic bacteria. So there this was a very common practice of using pulse filled gel electrophoresis. So now let us understand the first one, that is the paper electrophoresis, because this method was developed by Elvis, who is the father of electrophoresis. So though this particular technique is not much used, but in order to respect the first method of electrophoresis, I would like to share some views on this. So this particular method, we use paper strip or cellulose acetate membrane, okay, as a supporting medium. So this particular method, it is used for separation of small charged molecules like amino acids and proteins, okay, using the paper strip. So these are some of the methods which are used. So paper, it mostly contains 95% of the cellulose. So this but the problem with cellulose is that it is a poor conductor of electricity. Secondly, it is not transparent. So we cannot see. Okay. The next is it is going to absorb the protein. So it is going to give you bad results, or what we call as the poor results. So now different varieties or the grades of filter papers like the Whatman paper are available, which are specifically designed for this. Then next is cellulose acetate. Okay, cellulose acetate is a non-absorbing group. So previous one, that was cellulose, was a absorbing group. Now whereas the cellulose acetate is a non-absorbing group. So it gives comparatively better results, and moreover it is bit transparent and compounds can be separated easily. So this is also mostly used for clinical studies. Then next, what can be used for this is your starch. Okay, it forms opaque gels on boiling and cooling. So it is another non-absorbing and free of impurities. It gives better results of especially for isoenzymes. Okay, but always the favorite one has been the agarose one. Okay, so we will see this later on. So first let us understand some of the concepts involved in the paper electrophoresis. So in paper electrophoresis also what is done is, if you see this particular picture, okay, if you see this particular picture over here, you can see very easily, you can see there is one tank over here, you can see one anode and a cathode, and if you see it is having place for a buffer reservoir where we have the buffers. So we are going to place the paper strip over here and we have a wick. Okay, so normally we make a filter paper wick and it is kept in contact with the paper strip as well as the buffer. So because of the capillary action, the buffer is going to move or it is going to pass through the paper with the help of the wick or the filter paper wick that we have used. So there is continuous movement of the buffer which do not allow the paper to dry. So this is the setup involved over here. Okay. So here also the principle remains the same. If a direct current is passed through a wet paper containing the charged molecule, the positively charged cations, they will start moving towards the cathode, right? And the negatively charged anions, they will start moving towards the anode. So the mobility, okay, the mobility of the molecules will depend on the shape of the molecule, the pH, okay, sorry, and the composition of the supporting medium, okay, and the applied current between the electrodes. So it does not require a separate matrix preparation, and also it does not contain charge, okay, which is going to interfere with the separation. So spots, we load the sample in form of spots. Okay, now suppose this is a membrane that we have. Okay, suppose this is the acetate paper. So what we will do is we will spot the whatever the product we want to separate with the help of the micropipette. Okay, the same way how we do it for your paper chromatography. So same way we are going to spot, and these spots they migrate according to the charges. So after electrophoresis is done, the separated components can be detected using some special staining techniques, okay, depending on their chemical composition. So this is about the paper electrophoresis. So procedure also we have discussed in short. So coming to the application of this particular paper electrophoresis. Now first of all, it is used for distinguishing solutions which contain micromolecules in different proportions. Then nextly, to understand the change in charge of the molecules related to pH change. So how it is affecting. The next is separation of small biomolecules which are present in the solution. Then if we are using a lower voltage paper electrophoresis, it can be used for separation of proteins, carbohydrates and nucleic acids. High voltage paper electrophoresis, it is specially used for separation of amino acids present in a mixture. So this was all about your paper electrophoresis. But before we move on, so I would like to discuss some of the disadvantage of this particular paper electrophoresis. Now problem with this is it is low resolution. It is going to give you a low resolution result which is not acceptable at a scientific level, because especially these techniques are used in your laboratory, and people who are working in laboratory, they are looking for some results, right? So this results, it may be with the viewpoint of some work they are doing and which they want to publish. So definitely you need to give good resolution images, good resolution results. Or if you are working for some development of some product, so definitely you need to be very sure of whatever you have separated. So such a thing, it becomes a problem because of the low resolution produced. Secondly, it becomes little laborious um, action, that it is comparatively slower as compared to the gel electrophoresis process. Alright. So with this, let us move on to the main content, that is the gel electrophoresis. So gel electrophoresis, why it is so different from the paper electrophoresis? That is because of the use of this agarose gel. So we use the agarose gel, okay, for separation. Now what is done is, okay, this entire process, it depends on the separation of molecules based on their charges and size, using agarose medium as the supporting medium. So you must have seen this one program, that was Takeshi's Castle. I must have given this example long back. Okay, let me show you just to recall the one game called as the honeycomb maze. So where the maze was arranged in form of a honeycomb. So there were hexagonal cabinets. So a person has to move inside, and without getting caught, they had to pass through the doors. So actually your electrophoresis is somewhat like honeycomb maze only. So what happens here is, so this is the example of that. You must have seen this. So a person had to, so person has to move from these doors and pass. So finally that person has to reach the destination. Suppose they have moved here. So wherever the door is present, they have to move out. This is what happens in case of your electrophoresis. Now suppose if we have these, or if we make these hexagons or honeycombs of different sizes, okay, and if we have people also of different sizes, suppose if the door is very small, and if we ask some heavy person to move through, that person will not be able to move. So even if he is trying to move, it will take a lot of time for him to pass through, right? So same thing happens to your DNA molecules. So if the DNA molecule is small, it will pass through this pores very easily, and it will reach the endpoint first. Whereas if some DNA molecule is hefty or large molecules, it will be very difficult for that molecule to pass through. So it will try to move from wherever the place is available. So that means it will take more time to cover the same distance. So DNA gel electrophoresis, you have to remember this particular honeycomb maze. So it will be very easy for you to understand the principle of this particular topic. Just clear this off for you. Okay. So what is done is, we have a medium, that is gel. Okay, that gel is agarose gel. We will see what is that in bit of a moment. So we have this gel made over here, and this is the setup. So just like your paper electrophoresis, we will have a tank which will contain the buffer, and here we are going to place the tray or the casting tray, or sometimes it is the gel is directly slipped onto the surface over here. The electrodes are connected based on the charge of the molecule. And if you see here some dotted lines, okay, which are present over here also, the wells, they are the depressions, okay. They are made with some special tool called as a comb, which looks like a hair comb. So here we are going to load the sample, okay. Here we are going to load the DNA, and since DNA is negatively charged, okay, here we will have a positively charged electron, and we will pass the electric current with the help of through the buffers. So what will happen is, these DNA mixture, they are going to separate. So the smallest one is going to reach the end point faster as compared to the largest one. And in between, based on the size or variation of the size, we will have the separation of the electron. of the DNA bands. So this forms the basic principle of gel electrophoresis. Yes. So here also the principle remains the same. So if a direct current is passed through an block of agarose gel, okay, which contains the charged molecules, the positively charged cation will migrate towards the cathode and a negatively charged anion will move towards the anode. Now, if the question is asked in particularly about the DNA, you have to talk about the negative nature of the DNA. So, negative DNA will start moving or it will migrate towards the positively charged end. So this is a point that you have to remember. Then mobility of the molecules also it depends on the net charge, size, shape of the molecule. Also the pH composition of the medium and the buffer. The electric current that has been passed. So this agarose, okay, what we are talking about. This agarose is extracted from a seaweed. It is extracted from a seaweed and it is present in form of some fine white powder. This powder, what is done is it is melted in a buffer and it is heated for proper distribution. So during this time, what happens is this agarose it forms a gel by formation of hydrogen bonding. Okay, which contain and now this hard after cooling it forms like your jelly. Okay, that's what you eat. Okay, so it contains the pores. And through this pores, your DNA is going to sieve through or it will move through. Okay? So it is normally obtained from a red algae. Chemically talking about your agarose, okay, it is a linear polymer which is made up of D galactose and L galactose. So it's a polymer made up of D and dextro and levo galactose. This you have to mention in your answers. So when it is mixed with water, as we have spoken, there is cross-linking between the agarose units, the two units what we have, and it is going to form the porous matrix. So if a solution is dispensed within the gels, the pores, it is going to have a sieving capacity. How we have a sponge. If you see a sponge, piece of sponge, we will see there are minute pores there, which will absorb the water, right? So same way, here also there will be minute pores form, which gives the sieving effect. So as a result, the smaller molecules, they are going to move towards the anode faster as compared to the larger molecules. So pore size is dependent on the concentration of your agarose. So normally what we use is 0.8%. Okay. So most of the experiments, mostly we use between 0.8 to 1% of agarose gel. So then we have to standardize the method based on the study that we are conducting. So if we know the composition of your product very well, then we can determine the concentration of agarose to be used. But mostly it is like between 0.8 to 1%. Now see, if there is less of agarose, what will happen? So forget agarose. Let's take example of custard. So you must have used or you must have, some of you must have who like cooking, they must have prepared custard. So if you make more or if you put more of custard powder in the milk, what will happen? It becomes thick. Correct. And if you add less of custard powder and when you boil it, it still remains like a slurry, thin, right? That is what the difference remains over here. So if you put more of agarose, so for example, if we are using 8% agarose, sorry, 0.8% agarose. So 0.8% agarose will contain zero point 0.8 grams of agarose, which we will add to 100 ml of water. So sometimes it is made in buffer, sometimes it is made in distilled water. So we are going to use zero point 0.8 grams, we are going to weigh and we are going to transfer it in the uh, water and shake and boil it, right? And same way, if we want to prepare 1%, so we will take 1 gram of agarose, add to 100 ml and mix it. This will be more, it will have more compact nature as compared to your pointed graph. So again, it will depend what is the nature and what is the difference in size between the molecules that you want to separate out. Let's see what are the factors that affect gel electrophoresis. So point number one is that what is the charge on that particular molecule that we want to study. Next is what is the molecular weight. Then next is this if there is any sort of secondary structure involved, because that is going to affect the shape of external environment, the pH of the solution, the electric field, that means how much charge you are going to apply, the viscosity of the solution and the temperature. Because if your temperature is very high, it is going to melt the gel and we will get something called as the smiling effect. Because these wells, what you see here, they are like lines, right? So rather than that, we will start getting like this smile. So it is called a smiling effect. That smiling effect occurs when your temperature is more or your entire pH system is heated more because of the electricity, that electric charge that you have passed. So these are some of the practical mistakes which we actually do in the lab. So once I was working with one of my lab mates. So we face a similar problem, and instead of getting this intact separation of bands, we got something like smiles. So though it was not our mistake, it was because of the fluctuation of some electric issue was there. So we had to repeat the experiment. So these are some of the factors which affect the gel electrophoresis. Now let us see what are the things which we use for your gel electrophoresis. The first one is the gel. So as we know, what we use is the agarose gel. Then depending on the experiment and the difference in the molecules that we need to separate, we will determine the composition or the percentage of the agarose gel that we want to use. So this is specially used for nucleic acids. So proteins we will discuss about it later, but it is exclusively used for nucleic acid separation. So what is done is we take agarose, we are going to weigh it, okay, how much it is, okay, whatever we want. Suppose if we want 0.8% or we want 1%, we will see, we will weigh it, we will add to the suitable buffer or distilled water. Now, there are some labs, what they do is they add one particular chemical to it so that it can be visualized. So, we will come to that later on. We will explain it later. So, as of now, we will say that it has been boiled, agarose has been boiled so that the agarose powder is completely dissolved with the distilled water or the buffer. And then we have something called as a casting tray. If you see here, it is like a bench. So here we have a protective wall and here also we have a protective wall with a depression here. If you see here, there is a depression. So here also we have a depression so that we can use or we can make two lines. So what is done is normally in our Indian labs, so we will put some masking over here in form of a tape so that when we pour this gel, it remains intact over here. Okay. So when the gel is still in the liquid form, we are going to insert a comb over here. Okay, we will insert a comb. Now this comb will have the tooth or the teeth, the way we have for our hairpins. This is inserted over here and here. So what happens is as it hardens, it will form that depressions. When we remove the comb very carefully, we will see there will be some depressions here. Okay, if you see here, these are the depressions which are called as the wells. And this is the place where we are going to load the sample with the help of our pipette or the micro pipettes. Okay. And now if you see microscopically, okay, we will have something of this sort of look. The gel what we have, it will have something of this appearance. And if you see, there are some pores here of different size. Depending on the size of your DNA or whatever nucleic acid we are separating, it will pass through this and it will reach the destination. So this is the nature of the gel. So next, how we are going to visualize it. So for that, most commonly what is used here is ethidium bromide. So this is one cancerous component because it gets stacked between the base pairs. Okay, it gets stacked between the base pairs and by the process of intercalation. So if we are using ethidium bromide for prolonged time, it may affect our health. So it is always advisable to use hand gloves, good quality hand gloves while working with ethidium bromide. Then secondly, the thing that I have learned in my practical experience is that it is always better to keep one dedicated glassware in form of a beaker or a flask, okay, to use this or for boiling the gel. Because when we are boiling, as I had mentioned in the previous slide, we have to add some component here. So that component what we have to add to this boiled agarose is the ethidium bromide. So this we have to add and stir it nicely so that it is properly distributed. So it is always advisable that we keep one dedicated, one special flask or some glassware for the use of ethidium bromide. Because if we use it everywhere, it may get absorbed and it will affect the person who is handling it. Because when we are working, for example, for a PhD, you are working for years there, right? You are getting exposed to it very continuously. So it may affect you, which we may not understand initially, but somewhere in later point of time, we will understand this. Sodium bromide. Okay. How it works? It is very simple. The principle here is the ethidium bromide that what we have, it is an intercalating agent. Now suppose these are the base pairs present, okay? Suppose now these are the base pairs. Your ethidium bromide is going to be present over here. That is called as the intercalation. It will get stacked between the base pairs of the DNA. And when it is exposed, so after your process is done, your entire electrophoresis is done, we keep the gel on the UV trans illuminator, where it will get exposed to UV light, and we see, here if you see here the orange color fluorescent bands, that is what we see the ethidium bromide, which is intercalated between the base pairs of the DNA, okay, in form of the orangish fluorescent band under the influence of UV. Now why this happens? This is because the electrons which are present in the ethidium bromide, that is the aromatic ring of ethidium bromide, they get excited or activated and they will release the energy in form of light, which is visualized in form of bright orange color fluorescent bands. Now how much we prepare? So normally the stock is prepared as 10 mg per ml and the working concentration is 0.5 microgram per ml. So this is the thing that we have to remember. Because again then each lab will have their own standardized procedures. So that will again differ based on this. So what are the different alternatives we have? Because since ethidium bromide, it has health hazards associated with it, but we have this um, SY bromide, okay, gold, then SY bromide green, crystal violet, methyl blue. These are some of the components that we have. But mostly, even till date, we prefer using this ethidium bromide because there are many economical issues also involved there and many other things. Now this, sorry, methyl blue what we have, it is not, or crystal violet, they are not going to affect your health, okay? They do not secondly, they do not require any sort of UV exposure or any sort of light, okay, because they can be used easily. But apart from this, still there are problems associated with it. Okay. Next, we will talk about the loading dies. Now, in the previous picture, you must have seen here. Let me clear it first. You must have seen this blue colored thing, right? So we have to mix a loading die along with the DNA because everything is transparent. How we are going to see where our DNA has reached? Because by the time we expose it to DNA UV light, it is very likely that your DNA must have ran away, right? Must have ran or it must have got back into the buffer. So this is like use of this tracking die becomes very easier, very essential. So we also call it as a loading die or tracking die. So what is the work of this loading die or tracking die? So if you see, the wells are like this, right? And the buffer is flowing through this. So when we load the sample here, the DNA, it should be able to stay inside the well rather than moving out of it. So that is of no point. So the main function of this loading die is to make it settle down. And second function is when we are running the gel, okay, we have to see when to stop because everything is moving, right? So it should be done in such a way that somewhere, the loading die should run faster than the DNA. So it is normally said that we have to make sure that the DNA is running the or covering the 3/4, okay, 3/4 of the distance. Then we stop and then we visualize. If at all is, if we do not get good resolution, we may run it for some more time. So if you think logically, a loading die should have two functions. One is to give the density so that the DNA molecule settles down in the well, and secondly, it should provide the color so that we know when to stop the gel running of the electrophoresis. So the dyes that move at a steady rate, in a gel, they can be used. It is used for understanding how far it has reached. So normally we have a different compositions of loading dye or the tracking dye, but most commonly that we use is this. That is what I was using. Sucrose and xylene cyanol or your bromophenol blue or this ficol and orangey. So I had some of my teachers who used to call it ficol and some of the teachers they call it ficol. So I got fixed with ficol. So you see what your teachers are calling it and decide. So then another one is glycerol and bromophenol blue. So these are some of the compositions. So if you see, there are two components involved over here. One will provide the function of density and second one will give the color. So ficol is going to give the density, orangey it is going to give the color. Sucrose is again it will give the density and xylene cyanol or bromophenol blue will give the color. So we have this glycerol, again it is dependent or it is affecting the density and bromophenol blue will give the color. So this way we design the loading or the tracking die. So now let us go to the applications of DNA or the gel electrophoresis. So normally we know that it is a visualization technique. So it is used for specially for understanding or isolation of the DNA of any product like animal, plant, bacteria. Then it is used for studying the polymorphisms. So RFLP is restriction fragment length polymorphism. Then it is used for studying the paternity test, DNA fingerprinting. Okay, it has played a major role in the development of human genome projects. So it is used for understanding human genomics, bioinformatics, restriction studies, digestion studies. It is also used for understanding the infectious diseases, okay, to screen the clone products. Okay, after PCR, we use electrogel phoresis. Then it is used for variety of diagnostic studies too. So these are some of the applications of DNA electrophoresis. So with this, we have finished, or I have tried to make you all understand what is gel electrophoresis And this is like what I have spoken about. It will help you a lot in your practical Viva questions, because normally the examiners, they like to question you whether you have understood the each step and purpose of each chemical that is used. So for my TY exams of my B.Sc, so that time I still remember I was questioned on the composition of the trekking done. So very luckily, just before entering the practical exam, I had studied it and gone, so I was able to answer it. So till then, keep studying and keep smiling. So if you want to join any of our courses, please check the captions below and follow us for more details. Thank you.